Okay, so I'm speaking to you today about NAP, davunitide, an experimental compound which has received orphan drug and pediatric rare disease designation from the FDA, displays effectiveness in treating symptoms of autism and Alzheimer's disease. And I started speaking about ADNP. So about 20 years ago, we discovered it, and some of you heard me, so maybe I'm repeating myself. We discovered a new protein to science, and we called it activity-dependent neuroprotective protein, or in short, ADNP, because it protects nerve cells against electrical blockade. And soon thereafter, we discovered that it interacts with a very famous, to me at least, chromatin remodeler called SWISNF, which is actually associated with the serine coffin syndrome that we've heard about. And what we also found is that when we knocked out ADNP completely, there is no brain, so it's a no-brainer. Without ADNP, there is no brain. Only heterozygous mice live. And in children, if there is a heterozygous mutation in ADNP, and that was discovered many years later, then the child is within the autism syndrome, autism spectrum disorders, and ADNP is one of the leader genes among about 15 leader genes. And at the same time, when we discovered ADNP, we looked to see if there are active motifs in ADNP that maybe can also mimic the neuroprotective activity that ADNP shows against electrical blockade. And we discovered a very short peptide that we called NAP, NAPV-SIPQ, that protects against electrical blockade. And just from an entrepreneurship point of view, so we have a novel disease target, which is the ADNP syndrome, and we have proven safety and efficacy of NAP or davunitide in human trials, because it had been previously in human trials in in prodromal Alzheimer's disease and also in schizophrenia. And we have also some FDA perks like an orphan drug designation and the pediatric rare disease designation for the ADNP syndrome. Here is a child with the ADNP syndrome. She carries the most abundant mutation in ADNP, p-tire 719. She developed quite slowly, and what was obvious is that the motor development was very impaired. She's now 15 and she doesn't talk yet. And so there are many problems to the ADNP children, and I enumerated them here. And also, there are very many different mutations. So each child has a different as I said, there are some prevalent mutations, but many of the children have different mutations. Regardless, the phenotype is not that different. They are all delayed in development. They all have motor problems. Many of them do not talk, so they have intellectual disabilities, and many of them are show autistic. All of them show autistic traits, and many of them are defined as autistic. And ADNP interacts with many other genes and regulates during development more than 600 genes or 500 genes. And because of the discovery of mutations in ADNP in autism, de novo mutations, so it's not inherited, it is in the child, in the embryo, I decided to look also and see whether there is such a mutation also in the aging brain, whether it is something to do only with the developmental disorder, or is there a connection to other diseases. And we looked for mutations in postmortem Alzheimer's brain, and we discovered mutations in ADNP and in many other genes. But what is interesting, and I want to bring you back to a work that was done here in Ben-Gurion University by Professor Idan Menashe, he looked to see what are the genes that are most prone to mutations in autism. I hope I'm saying it correctly. And he found that it's very long genes that are, and ADNP and SHANK3, for example, are among these, and CHD8, I think, are among these very prominent single gene mutations that are occurring de novo. And the question is that only for autism, or maybe it's also can occur somatically? And indeed, we discovered that many genes that are associated with cytoskeleton are somatically mutated in Alzheimer's disease and in the aging brain. And then there were two other works at the same time that discovered other genes and similar genes, but then a fourth work, looked at the three work together and found the same finding that Idan had quite a number of years ago, that it's large genes that undergo somatic mutations also in the Alzheimer's brain. So there is a connection between Alzheimer's and autism, and the connection is also that the mutation in ADNP increase with increased tauopathy. So you know, the Alzheimer's brain has two major protein pathologies, the amyloid plaques and the neurofibrillary tangles that are made of the protein tau. And the protein tau is a microtubule-associated protein that is very important for neural outgrowth, for axonal transport, and for synapse formation, etc. And what I'm connecting here is the disruption of microtubules and disruption and tau deposition is associated very closely with Alzheimer's disease. And what I want to show today is it's very closely associated also with the ADNP syndrome and probably many other autistic syndromes. So here is actually Idan's work in the side, and here is another work showing that ADNP is one of the leader genes. This is one of the recent work showing that ADNP is one of the leader genes in neurodevelopmental disorders. And what I'm showing you here also is that in our model of ADNP haploinsufficiency or heterozygosity, when we look at dendritic spines, you see how nice there are and how many there are dendritic spines here in the mouse brain, where it has intact ADNP. And then when it loses all ADNP, there is no brain. When it loses one copy of the ADNP gene, then the dendritic spine is kind of stunted, and there is not so much growth. And then when we treat the mice for 9 days by injection, by intraperitoneal injection of NAP, the small fragment form ADNP, we actually can recuperate the dendritic spine. And so the haploinsufficient mice that we have, the heterozygous mice, show the same problems as the children, and we can repair by NAP treatment. And this was published 2 or 3 years ago in JCI, and I think I've spoken here at the first meeting about this paper. Now we have a new paper, and we have a new model with the most prevalent ADNP mutation. That's the p-tile 719. It cuts the protein not to half, but almost, and it actually gets rid of the nuclear localization signal and also of the homobox domain that there is in ADNP. So there is actually, when I look today at the ultrasound picture and the differences in structure of the brain, this might be associated because many autism genes have homobox domain and are associated with the formation, like ADNP, that associated with the formation of structure. And also when we look at the postsynaptic, the presynaptic side, and we look at glutamatergic transmission, what you see here is that in the intact mouse, you see a beautiful staining of the glutamatergic neurons, and then when we knock down haploinsufficient ADNP, then we see a dramatic reduction, and this is in the hippocampus, and when we treat with NAP, we can see the recuperation, and this is comparing to the child brain that there is a problem in development of the hippocampus. So the hippocampus is not developing properly in the ADNP children, hence probably are the cognitive impairment or the intellectual disability. So how does it work from a biochemist point of view? And I started my career as a biochemist. I mean, I did my PhD, the department was called biochemistry, and then it turned to neurobiology. So I have a PhD in neurobiology, but I started in biochemistry. So anyways, this is irrelevant. What we discovered is that NAP actually has the structure, the signature structure of binding to microtubule and binding protein. So, you know, microtubules, without microtubules, we don't have a neuronal structure. And we have the microtubule and binding protein that binds to the end of the microtubules and then make the microtubule dynamic. And you need this structure in order for the microtubule to move and for axonal transport to occur and for synapse formation. And as you see here, it's like a key in the keyhole, and the structure of NAP sits on the microtubule and binding protein and make the microtubule move. Oops. Sorry, just 1 second. And here you see the movement in red, and you see that with NAP, the movement is faster. So NAP enters the cell through a dynamine-associated endocytosis, and then it interacts with the microtubules and makes the move. So you need ADNP for the movement of the microtubules, which is very critical. And when there is an ADNP mutation, the microtubules become shorter, and this is what you see here. And you see the different mutations, you see much shorter microtubules, and when we treat these cells with NAP, we actually get the length and the mobility is similar to the normal situation. And what we think is happening also is that microtubule end-binding protein interacts with the protein tau, remember the one that deposits in Alzheimer's disease, and actually enhance tau binding to the microtubule. And when we incubate them in the presence of NAP, we actually get 20-fold increase in tau microtubule interaction. And so the next question was, can we see tauopathy, like in Alzheimer's disease, also in a post-mortem brains, like from ADNP children? Unfortunately, there were some deaths already among the not so many ADNP children are there in the world, known to men, there are about 400, but there were already some deaths, not so many. And we got post-mortem material, the parents wanted us to look at it, and we looked at it, and also the clinicians. And what we saw is we saw tauopathy, and instead of my finger, I hope you are seeing, I don't know if you see, no, you don't see my cursor. So you see here the deposits are actually labeled with green arrows, and you see the tau deposits, and you see it, this is in post-mortem from a child 7 years old, and you see, like here, the control is actually a 30-year-old person, and you see that there is no tauopathy whatsoever, and the child has tauopathy. And tauopathy is associated with cell death, with inhibited axonal transport, and maybe also with accelerated aging, but this I don't know if I can say. So what you see here is also, in cell culture, we are looking at tau microtubule interaction, and you see that the tau microtubule interaction is reduced dramatically if there is a mutation in ADNP. You see the mutation that I'm all time talking about, the p-tyrosine 719, and you see correction in the tissue culture by NAP treatment in orange. And those of you who have seen poster number 2 saw this, and we have just published a paper on our new ADNP model that has a mutation that we created by CRISPR-Cas. This is the PhD work of now Dr. Gidon Carmon. And we characterized this mouse model, and I wanted just to bring to your attention. So we looked, remember there was a talk in this hall, the first talk by Marina, and she was speaking about vocalization, and we also measured vocalization in the mice, and the syllable emission by the mice, and this is ultrasonic, and we see that the tyrosine, we call them tyrosine mice, so the ADNP tyrosine mutation mice, they show a much less rich syntax or poorer syntax, and then when we treat them with NAP, we actually can increase enrich the syntax, and they behave normally. And also we looked at the mice from the birth, and we saw actually that there is some death just after birth. We saw inhibition of eye opening, later eye opening, and we can correct it by NAP treatment. We saw developmental milestone inhibition, and we can correct it by NAP treatment. And we also saw, we measured gait. So as I said, the children have delayed motor development, and we measured gait, and we saw development, and you can give me 10 because you gave the other one 20. No. And we also, okay, and we also by CRISPR-Cas, we showed that if we reduce ADNP only in the muscle, we inhibit the gait, and we can correct it by NAP treatment. So it's not only central nervous system, but it can be also directly in the muscle. So having ADNP child or having an autistic child with ADNP mutation is not only an effect of on the brain, but also, for example, on the muscles. And the other finding that we had in these mice with ADNP mutation is that they have tauopathy, as you see here in the picture. You see the brown stain, and then we can reduce it by NAP treatment dramatically. And also we have impaired visual evoked potential that we can correct by NAP. And the blue is the impairment, and then the green and the red are the control and the correction. And we saw major changes in gene expression that were sex-specific. And the major protein that we have seen changing, a major protein, is FOXO3. And FOXO3 is a transcription factor that is associated with regulation of the microbiome, which I heard today that is very interesting for the SFARI Foundation. And also, FOXO3 is one of the major genes and proteins that protects us for healthy aging. And so ADNP regulates this gene. It also regulates the microbiome, and this is poster number 1 that Alexandra was showing you today. And here we show that the same genes that are regulated in the mouse are also regulated in lymphoblastoid cells from different mutated patients. And lastly, I also wanted to mention another work another recent work. So FOXO3 that is associated directly with healthy aging also interacts with another protein that is associated with healthy aging that is called SIR2-in-1. And SIR2-in-1 actually protects against tauopathy that we've spoken about. And we have shown that ADNP and SIR2-in-1 interact. We've shown it in iPSC cells, actually with Gad Batin, Professor Gad Batin from Ben-Gurion University. And we've shown the direct interaction with microtubule end-binding protein. And Maram Ganaim, who sits here in the audience, she actually did these beautiful gels. And this is the interaction. So ADNP interacts with SIRT both on the microtubules, enhancing tau binding to the microtubule, which is further enhanced by NAP treatment, and also at the chromatin level, as you see here, and disruption of this, like in Alzheimer's brains or in Parkinson's brain, causes a complete disruption of many proteins that are associated with chromatin epigenetics. And just as the last point, NAP, or as we call it also davunetide, had been in clinical trials. And this is, as I said, and this is in amnestic mild cognitive impairment patients, so this is not such healthy aging. And NAP protected memory, and this is associated with short-term memory, remembering structures that you have seen. After 12, you've seen a structure, and after 12 seconds, you see the same structure with 3 others, and then you have to differentiate. And you see here a dose-dependent protection by NAP or davunetide treatment over 12 weeks treatment, daily or twice daily treatment, and then cessation of treatment, and the effect was maintained for another 4 weeks. And the same similar results were also discovered in schizophrenia. And this was done by leaders, psychiatrists in the United States. And what they saw is an effect on activities of daily living. And again, there was a significant effect of davunetide or NAP treatment. So I told you about the regulatory achievements, and I hope that we will go to the clinic pretty rapidly. And I thank my laboratory members for their great help, and there are many more. I had like 65 students over my career, and now my students are here, and I mentioned them. So, and these are my prizes, and this is the end, and I'm happy to answer questions. And I think I